Thanks so much, Gordon, and I'm delighted and really privileged to be speaking with everybody here today. And I just want to endorse everything that Gordon has said about the value of HRECs and HREC members. You all do a magnificent job. My task today is, I think, to talk about one of the most challenging areas that HRECs have to deal with, and that is genomics, because it's kind of complex. The science is complex, and the ethical issues are, of course, complex as well. So I do have slides, and I will get myself organized to share those slides. Hopefully this is all okay.
Yep, I see perfectly. Thanks, Di.
Good, thank you. Yeah. So thanks, Gordon and the team, for the invitation. As I said, I'm really privileged to be here today. I must say it's quite unusual for me to have so long to talk, and it was interesting in preparing, thinking, oh, I have all this time, but of course I'm probably going to run out of time, so I'll do my best anyway to keep on track. And in preparing for this presentation, it also really struck me that I would be talking to a very diverse audience. I'm sure there's some of you who are fully across the ins and outs of assessing applications for ethics approval for research that involves the generation of genomic data, but others will not be. And so what I'm trying to do is sort of give you a little bit of a background to help flesh out some of the details and think about some of the issues that come up for HRECs in this area. And I'll come back to an issue that's come up in a recent publication to do with confidence in genomics and what HREC member attitudes are towards this. It's really interesting. This research came out in the European Journal of Human Genomics a couple of days ago. So my plan for today is first of all to go through some scene setting. I ask the question, why genomics? Why genomic data sharing? And I'll talk a little bit about my research group, some of the critical terminologies, and then the big research question in this area that's referred to as ELSI, or ethical, legal, and social implications. And then go on and talk about the changing nature of genomic research and the changing environment in where it's situated. And then we'll turn to issues for Australian researchers and HRECs. I'll talk about genomic confidence, some of the key publications that guide my evaluation of this area, and then three big issues that I think are some of the core issues for HRECs to consider: consent and waiver of consent, privacy and anonymity. And Gordon's already said that you've heard something about that already. And I think you also heard about the concept of dynamic consent yesterday as well. And then finally, I'll turn to return of research findings. So it's a big job, but let's go ahead. So the first bit is why genomics and why genomic data sharing. And I want to introduce you to my team at the Centre for Law and Genetics. So I've been director for our centre since 2015 and a member since its formation in 1994. And my group and I are really interested in many of these ethical, legal, and social implications, or ELSI, of genomics and genetics. And don't worry, I'll come back to those terms in a moment. Recognising that these ELSI are global, it's important that we work within the global context, but it's also, I think, really important that we examine these issues through a distinctly Australian lens so that they're locally as well as globally relevant. So as a team, we're invested in this research, but we also have broad experience in research ethics governance as well. So I've sat on an HREC and I chaired an animal ethics committee. I've been on HREC and I'm currently chair of the NHMRC Embryo Research Licensing Committee. My colleagues, Don Chalmers, might be familiar to you. He's been in this area for so long and was chair of HREC in the 1990s. Margot Losky was on HREC for a number of years and has chaired one of our local HRECs. Lisa Eckstein is the immediate past chair of the Tasmanian Human Research Ethics Committee. And our other colleagues, Jane Nielsen and Jenny Caldor and Beck McWhirter, have also been involved in research ethics for a long period of time. I also want to pay special attention for my dear colleague, Christine Critchley. Christine was at Swinburne University and a social psychologist, and she did some incredible work focusing on public trust in genomics and how public trust should be used to help us guide research ethics and regulation. Christine tragically passed away last year, and we dearly miss her, but we just still value her contribution to this field so much. And I'll be talking a little bit about some of her research in a moment. I just want to mention that research in the presentation today is funded by the Australian Research Council through a discovery project, Issues in Law, Research Ethics and Society relating to genomic data sharing. And I'm also going to talk about contract research that we've been doing for the Department of Health on the legal and regulatory landscape for genomic data collection use in Australia. But I really do need to make this disclaimer that the views are entirely my own or those of my research team, and not in any way views of the ARC or the department. Okay, then, on to terminology. So you'll see that we are called the Centre for Law and Genetics. And back when we started our research, genes and genetics were still words that were commonly used in the field. The concept of genomics came later, and I want to talk a little bit about how that came apart. The Global Human Genome Project had just commenced when we started. This was one of the first big international science projects. Governments in various countries agreed to fund this ambitious project, which aimed to map all of our genes and work out the sequence of all of our DNA, our whole genome sequence. By sequence, what I mean is the precise configuration of the series of nucleotides, the bits of DNA that make up the whole of the sequence, the necessary information that allows us to develop into human beings and maintain ourselves throughout our lives. Sometimes you'll see reference to the whole exome sequence as well. This is the components of the human genome that are actually used in our cells to make proteins that allow us to function as human beings. But there's a lot more to the human genome than just our genes, and I'll come back to that in a minute. So the funding commitment for the Human Genome Project was huge, but it was a gamble that paid off. By 2001, gene locations had been mapped, and there was a rough draft of the human genome sequence. The sequence, the final draft was published in 2003, and all of this was actually ahead of time and slightly under the budget of around $3 billion. But just to give you some historical context around this, the Human Genome Project began at a time when the internet was only just becoming mainstream. There was certainly no opportunity to interact online. People were only just starting to get email addresses and data processing. Sorry, I'm just checking a couple of chats that have come in, but we're all right. I'm assuming that everything's going okay.
Don't worry, Di, I'll deal with the questions, so you just carry on.
Okay, great. Yeah, thank you. And whole rooms were set aside for computer storage purposes, probably with as much memory as we now have on our smartphones. The analogy that was used for the storage problem was the equivalent number of telephone directories. And I'm sure many of you here today haven't even seen a telephone directory. The technical obstacles were also huge, which is why it took 13 years and $3 billion for the first sequence. The time and cost of sequencing has reduced dramatically. mere hours now and under $1,000 to get a human genome sequence. Of course, the cost of interpreting this massive information is still incredibly high. There were some surprises in all of this. The biggest one of which is we don't have hundreds of thousands of genes that were expected. There's still actually a lack of clarity around the precise number, but it's believed to be around 21,000 genes in the human genome. It was also found genes don't actually work in isolation, but they work together. It's erroneous to talk about the gene for. This is an oversimplification, even for the monogenetic diseases like cystic fibrosis and Huntington's disease. There's a whole sort of orchestra that is being conducted to get genes to function or to determine whether they don't function as we wish them to. It was also found that large patches of DNA, the patches outside the exome, are far from what was originally thought of as junk DNA. They too have important functions to play in gene variation and gene regulation. So it's complicated. And as a result, rather than talking about genetics, we talk about genomics, study of the structure and function of the whole genome. And we're still only just starting to understand genomics and how genomic and environmental factors interact together throughout our lives. Much more research is needed. We are starting to see some clinical benefits, including better diagnostics and predictive testing, therapeutics tailored to individual genomic attributes, and even some therapies aimed at modifying genetic defects. Indeed, the very first gene therapy for eye disease, a therapy called Luxturna, was recently approved by the Therapeutic Goods Administration. Of course, each of us, aside from twins or clones, have our own unique DNA. But the reality is about 99.9% of our sequences are common between us. And uncovering this common or reference genome wasn't an endpoint of the Human Genome Project, but it was really a starting point. So now what we want to do is understand how variation works, how variation in our sequence reflects on physical manifestations that make us different as human beings. So we need studies across whole populations. And these are often referred to as genome-wide association studies, or GWAS. And through GWAS, it's possible to associate good health and ill health within populations, within variations within the genome. Variations may actually be quite small, just single nucleotides, referred to as single nucleotide polymorphisms, or SNPs. And we do know there's some association between variations and good health and ill health, but we don't always know the significance of these variations. So there's variations of known and unknown significance. So we're starting now to see some of the issues with genomic data sharing that are relevant to this discussion. More research is needed, but we don't always know what research is needed. We don't know what it will reveal about participants. Genomic information is needed, but it often has to be linked with other health information to make it relevant. So this, of course, raises issues with understanding what people are consenting to, what they need to know, and how their privacy is protected. So the LC research question that I really want to focus on today is how we facilitate sharing of genomic data so that we can understand genomic variation while protecting this deeply personal and familial and sensitive information. So that's the task that lies ahead. And I want to give a little bit more context around this. it comes at a time when the landscape for genomic research and clinical activity is changing quite dramatically. And I've just drawn up two schemes here for the traditional view on research and clinical application of genomics. Here, hopefully you can see my arrow, you've got the individual, and they might be a participant in research, working in the research laboratory, or a patient in the clinic. And research findings might go into the clinic, the pharmaceutical industry, and the diagnostic industry, which all feed into the clinic. There's quite a bit of separation there. But in the modern world of genomics, the distinction between the participant and the patient is really breaking down. The individual is still at the center, but there's interactions between the individual and the clinic, and the individual and the research laboratory. The individual might donate material to a biobank, which is a repository for tissue and data that might be linked to health information to be used in many future research projects. Then we've got this peculiar direct-to-consumer testing industry that is going to be particularly relevant for our considerations. This is where consumers directly interact with testing facilities rather than going through researchers or their own GPs. And then we've got the diagnostics industry, pharmacogenomics and the like. It's all much more complicated. Similarly, the regulatory environment is changing. So the regulatory environment is complicated enough already. What I referred to rather whimsically as a regulatory soup, where we've got things like the duty of care owed in negligence law, duties of confidence, consent laws, intellectual property laws, consumer protection, professional codes, and so on and so forth. In this new environment, these are all breaking down. So rather than applying to specific sectors, they apply more broadly. So we're moving from a minestrone, if you like, to a consomme type soup. All of this adds to the regulatory challenges. To help us with our research in this area, on the changing nature of genomics and genomics data sharing, in our research, we identified 6 scenarios that are relevant where genomic data is generated and could be shared. And we developed these scenarios working with experts. So we interviewed a number of experts and pulled out case studies based on the information they were telling us and validated those case studies with other experts. And we've got 6 different scenarios here. First one is clinician-led, where a clinician is working with an individual patient and might want to share data with colleagues to assist in identification of the individual patient's condition. But then we might have clinician-led research where the clinician contributes data to a genome-wide association study to better understand how their patients' variations fit within population-wide variations. We might have a researcher-led project involving pre-existing genomic data, and we have to think about waiver of consent. Or a company-sponsored clinical trial. What happens to the data that's generated in the clinical trial? Or we might have a researcher-led project where data is being put into a biobank. And finally, citizen-led science, which is becoming much more common. This might be where a person engages with a direct-to-consumer genetic testing company and gets data which they share online with with their community for citizen-led science. So these points 2, 3, 4, and 5 are all relevant because they'll all require HREC approval in some way or another. So we'll be focusing a little bit on those scenarios. One of the things we've done in our research so far is we've looked at public attitudes towards genomic data sharing in each of these scenarios. And this was the work that Christine Critchley was involved in last year. She surveyed 243 people across Australia, asking them to identify and flesh out their views on preparedness to share data, to participate, what they saw as the rationale for participating and what worried them, and so on. And as you can see from this, I've just identified some of the key points that I thought would be of interest. First of all, there was a high level of interest in participating in sharing data. And this was what we've seen generally in our research. Australians are really positive about participating in genomic research in particular. A mean of 71%, ranging from 90 down to 30% for DTC. So less willingness to participate in direct consumer testing, but more positive in all sorts of other areas. The majority of people said that it was the potential benefits that drove them to participate. And it's really interesting. The benefits they identified were primarily general benefits to society, that their participation would help in the research effort, would help in subsequent development of diagnosis and treatment. It's much more unusual for people to identify personal benefits. And it really emphasizes that people participate in research for altruistic purposes. And we really need to identify and value this participation. However, a substantial minority did identify risks or negative consequences. And the sorts of things that worried them were privacy, data protection, commercialization, data going offshore and not being properly regulated. And you can see that the highest concerns related to DTC, lower concerns for participation in biobanking. And each of those themes relating to risks or negative consequence or the like changed depending on the scenario that the person was exposed to. So particularly risks and measures that would increase participation. There were particular concerns in relation to DTC primarily, but also participation in clinical trials and the like. So we do see some differences. So that gives you a little bit of background to what genomic data sharing research is all about and how the community sees it. What I want to turn to now is the second part of my presentation, what this raises in terms of issues for Australian researchers and human research ethics committees. And first, I want to turn to this issue of genomic confidence. And as I mentioned, there was a paper that was published a couple of days ago in the European Journal of Human Genetics. And it actually looks at an Australian study looking at Australian Human Research Ethics Committee members and their confidence in reviewing genomic research applications. You see a couple of my colleagues, Lisa Eckstein and Beck McWhirter, were part of this project. Some of you might even have participated in it. Interesting, what they found was their key finding was that there were low or moderate genomic confidence levels, even in HREC members who are highly familiar with genomic terms. Genomic confidence was, however, positively influenced by genomic education, familiarity with the terms that we've been discussing, and frequency of reviewing genomic studies. So what this illustrates to me is that we should not be too concerned if each of us, feels a little bit of anxiety about reviewing genomics applications. This is perfectly normal, and it's a really big task that HREC members are being asked to do in this field. And it's really important to think about how we might better assist HREC members in all of this. So there are many publications in this area, but there are three key publications, I think, that certainly guide me, and I think are really good guidance documents for you all in your consideration of these issues. Of course, the first one is the national statement. That goes without saying. I don't think I really need to say anything about that. But the second one is this report called Essentially Yours. This was a report of an inquiry that was undertaken by the Australian Law Reform Commission and Australian Health Ethics Committee. The government of the day in 2002 asked these bodies to look at the protection of human genetic information in Australia, listen to public views and views of experts, and give a report on what the key issues are. They published Essentially Yours in 2003, and it's 2 huge volumes of material. It's just a rich source of information. It was incredibly widely regarded at the time. It was the first of a comprehensive analysis of this area globally, and it still has resonance today, even though it's now 18 years old. The third document I want to mention is our National Health Genomics Policy Framework. This was developed in 2017, and it was a collaborative venture between the states and territories and the Commonwealth to set the agenda for health genomics into the future. So what I want to do now is just draw out a couple of things from Essentially Yours and from the National Health Genomics Framework. Essentially Yours had many recommendations, but to me, this is one of the ones that really resonates still. It says fundamental human dignity requires that individuals have a level of control over the genetic material. Human genetic information is personal and sensitive and deserves a high level of protection. So I think these are the guiding messages that come from Essentially Yours. The National Health Genomics Policy Framework provides a shared direction and commitment between governments to consistently and strategically integrate genomics into the Australian health system. And it has high-level application of policy, regulatory and investment decision-making for genomics. So this is something that governments are not shying away from. They recognise that genomics has to become part of the Australian health system. And they identify 5 strategic priorities. They emphasise that it has to be a person-central approach. Like the ALRC, it recognises that this is about people, and integrating genomics into healthcare requires this person-centred approach. It will require a skilled workforce that's literate in genomics, sustainable and strategic investment, appropriate services, and responsible collection, storage, use, and management of genomic data. So the governments across Australia are now looking at ways to integrate these strategic priorities. As part of this, they wanted to have a good indication of the regulatory and legal landscape within which this health framework had to be positioned. And so we at the Centre for Law and Genetics and our colleagues at the University of Melbourne, led by Professor Mark Taylor, were asked to do an examination of the regulation of health-related genomic samples and data within Australia. Essentially, it's an update of the regulatory analysis that was undertaken in Essentially Yours some 18 years ago. And there have been significant regulatory changes since then. There have also been significant technological changes, including the change from the language of genetics to the language of genomics. But it also recognises that the traditional regulatory frameworks based on the individual really fail to recognise the essential nature of genomic samples and data. They're inherently identifiable and shared with families, communities and populations, which is why we call our report essentially ours rather than essentially yours. And the report also recognises the traditional boundaries between the lab and the clinic are dissolving, as I've already mentioned. So we have identified a number of themes for legal and regulatory analysis. And the way we did this, we interviewed a whole range of stakeholders and did a literature review for the key aspects of all of this that we felt had to be looked at. Consent and waiver of consent is, of course, always going to be a major issue, as is privacy and its limitations. The issues associated with identity and genomics for our Aboriginal and Torres Strait Islander communities are, of course, absolutely critical. And it's really important to engage with Aboriginal and Torres Strait Islander scholars on this issue. Genomic discrimination is, of course, a pertinent consideration, as are controversial secondary uses, like uses for forensic purposes, uses for commercial purposes. Return of findings to participants is also important, as is the regulation of genetic tests and genetic testing and control over tissue samples and data. What I'm going to talk about specifically for the last 10 or 15 minutes are the issues associated with consent and waiver of consent for HRECs, privacy and its limitations, and return of findings. So let's turn to consent then. Consent, of course, does remain a central control over the collection and use of genetic samples and data in both clinical and research settings. But it's not the only thing that's relevant. Sometimes we hear people talking about having consent from participants, although as though that's an end in itself. A group of us got together internationally a couple of years ago to say, no, this isn't enough. Consent is insufficient for data release. And we had a letter published in Science that articulated our concerns in this regard. And then we went on to tease out some of the matters that are relevant when it comes to genomic data sharing, saying that we need better governance, more holistic governance of genomic data sharing. And trustworthy governance has become really one of the important catchwords. There are, of course, limits on the extent of consent due to questions associated with the appropriateness of broad or unspecified consent for future uses, consent for complicated or technical activities, and consent for activities with the potential to affect other people. So these are all issues that need to be considered when we're thinking of consent. And I just want to talk a little bit about considerations for future uses. Because as I've said, when we're looking at genomics, it's not just about one specific research project, but it might be, when genomic data is shared, it might be available for more than one project that isn't even thought about at the time that the person consents. So what happens when this proposed secondary use of genomic samples or data for research? When does waiver, for example, become applicable? So we've got this little schemata that we drew up for our Essentially Yours report. Just to go through it, we start up with proposed secondary uses. First question is this specific consent provided for the secondary use? If so, research may proceed subject to HREC approval. Or is there broad or unspecified consent proposed for future research uses? If so, it will be necessary for the HREC to assess to address the adequacy of the consent to cover research use. And then, of course, if it's adequate, research can proceed provided there's HREC approval. If it's not adequate, then the HREC will have to assess the availability of a waiver of consent. or if the original consent is silent on secondary uses, though it will be necessary to assess the availability of waiver. If it satisfies the waiver requirements, research may proceed, but if it doesn't satisfy the waiver requirements, reconsent may be necessary. So looking at waiver considerations then, there are a number of things, of course, you have to consider when you're thinking of waiving the consent requirements as set out in chapter 2.3, paragraph 10. I'm not going to go through all of these, but I've highlighted a few issues where the involvement in the research carries no more than low risk to participants, and the benefits justify any risk of harms. It's impracticable to obtain consent, and there's no known or likely reason for thinking that participants would not have consented. And then there are all sorts of other issues to consider, like protection of privacy and confidentiality, participants' welfare, role of commercialization, and what national and state and international laws say. There are some uncertainties then. Impracticability is not defined in the national statement, save that it says that it might apply due to the quantity, age, or accessibility of records. So HRECs have to decide for themselves what impracticability means. They also have to decide what no more than low risk means. The national statement does say that as a general principle, research, including genomics, will require HREC approval. But if there's no information that can identify an individual and no linkage of data, the research may be determined to carry low risk. And then what's the issue of presumed consent? How do you determine if there's no reason to think participants would not have consented? Again, no guidance. So this poses challenges for HRECs in making these determinations. There are other alternatives to waiver by HRECs. For example, England and Wales has a system where it's not the role of HRECs to make this determination, but there's a confidentiality advisory group, an independent statutory body established to do this, and it publishes its minutes. So that has the advantage of national consistency and a level of transparency. And there's some discussion about whether that might be appropriate in the Australian context as well. And then when it comes to privacy, we have similar issues. There have been a few studies that have come out globally that have shown that it's really difficult to say that genomic information can ever be said to be anonymized. It is inherently identifiable, and this has consequences for privacy law and practice. And as a consequence, claims of absolute protection of privacy are really unfounded in this area. There's also a problem by the sheer volume of privacy laws and the lack of consistency across Australia. We have a federal law, but most of the states and territories also have their own laws. So the federal law, for example, applies to federal government agencies and companies. But for example, the law in my jurisdiction, the Personal Information Protection Act, applies to state government agencies, including the University of Tasmania. So we've got challenges there for HRECs. I've just put this quote from Essentially Ours, which encapsulates some of the concerns we have about privacy. Privacy legislation is a key regulatory influence in the field, but its efficacy is hampered by jurisdictional inconsistencies and reliance on distinctions that are increasingly inappropriate, including the distinction between clinical and research uses, public and private, identifiable and non-identifiable information. This creates uncertainty for those working across jurisdictional or organizational lines. And then there's uncertainty as to what duties apply at common law, whether there is actually a tort of privacy or not. creates a complex national picture. One of the issues is whether genetic and genomic information is actually personal information protected by privacy laws. It's specifically mentioned in some legislation, but not all. Under federal legislation, personal information is defined as information or opinion about an individual, which is reasonably identifiable, which would seem to include re-identifiable information. Question is, does it cover genomic information where there's no intention to re-identify? Genetic information can also be health information and other forms of sensitive information. And then we've also got the Section 95 and 95A guidelines, which allow for waivers of Privacy Act consent requirements where information is coming from Commonwealth government agencies and private organisations. But there are differences when we're looking at state and territory legislation. So considerations, again, we've got this issue of impracticability coming up. Seeking consent is impracticable, and research activity cannot achieve its purpose through use of de-identified information. So again, another challenge for HRECs. So let's now go on to return of findings, and I'll quickly go through this because I know I'm running out of time a little bit. What are the obligations of data custodians, including individual researchers, to analyse and reanalyse data and communicate the findings to participants? There have been the recent amendments to the national statement relating to genomics, which help a little bit in this regard. What I've done here is set out the types of findings and the laws that apply. So there might be clinically relevant finding. A clinician asks for a genetic testing laboratory to undertake genetic analysis to identify, for example, whether there is a risk of heritable forms of breast cancer, for example. But there might be incidentally clinically relevant findings that aren't specifically looked for, but are clinically relevant. That we might have findings for these variants of unknown significance that I mentioned earlier. And these might become clinically relevant when they're reanalyzed. We've got the distinction between individual and cumulative research findings, and simply raw sequence data. Is there an obligation to return that? There are a number of regulatory instruments that are relevant. Privacy laws, national and international standards for accreditation of testing laboratories. The Therapeutic Goods Administration has requirements for in vitro diagnostic medical devices. Those are genetic tests. And then we've got HREC obligations and tort law. When we look at incidentally clinically relevant findings, I just want to talk for a moment about the US approach. The American College of Medical Genetics and Genomics has guidelines that they say there is a minimal list of about 50 variants that should be assessed by clinicians every time genomic analysis is undertaken because they have a high degree of clinical validity. And this is becoming part of mainstream genomic research as well. This hasn't been adopted in Australia, and I'm certainly not suggesting that it should be. The question, though, for us to think about is whether clinicians, researchers, and HRECs actually need more guidance on what should be undertaken. The national statement does have guidance, as I've said, on return of findings, and I'll just briefly go through these. It says for researchers to decide how results will be provided to participants, how the process will be managed, how risks will be assessed. And return depends on context. It says some must be returned, some may be returned, and some should not be returned. Doesn't say which information covers these areas, but things to consider are scientific and clinical validity, significance and clinical utility. So researchers and as a consequence, HRECs have to consider all of these things. It says there are no obligation to return raw research results, but where there are clinically significant findings, these can't be returned from the researcher directly, but must be validated by an accredited laboratory and must be accompanied by clinical and genetic counselling. Researchers do have a responsibility for recontacting if new findings come to light, but those end when the project ends. So it seems that there is a set of guidance there. And you'll be perhaps familiar with the decision tree. I'm not going to talk about that because it's kind of complicated, but I just want to draw out some key issues for the ethically defensible plan for return of findings. It says that researchers must address in detail whether findings will be returned, and if in the event they're returned, how will they be validated, how consent will be sought, how participants will be notified, how participant privacy will be protected. And this must be submitted to an HREC for approval. Gordon, I'm almost at the end. I've just got a minute or two. So issues. Detailed guidance is provided on the requirements researchers must comply with to secure ethics approval and the process that HRECs have to go through. But the questions that I pose is there enough guidance to researchers on what specific analyses should be undertaken and what findings should be returned? Is there enough guidance to HRECs in assessing the ethically defensible plan? And is there a risk of national inconsistency? A team has been working on the problems of international inconsistency and the problems this poses for ethics review and the need for ethics review mutual recognition. The idea that we need international standards, and if we've not got national consistency, we've got real problems in developing international standards. So with all of this, what we've seen is that genomics offers great promise for healthcare, but more research is needed within and across populations using shared data. It's occurring in a rapidly changing environment with dissolving boundaries between the clinic and the laboratory. People are generally positive about participation, but concerned about things like privacy, data security, commercialization. And ethics committees really face a number of challenges related to matters such as consent, privacy, and return of results. But having said this, it's clear people are not necessarily confident in engaging with genomics. So to me, I don't have solutions, but what we need is more conversations about genomics, more education, more discussion about harmonization of approaches nationally and internationally. And that's it, Gordon. I'm sorry for taking more than my massive allotted time to talk about these issues.
No, Di, when chair people go, that's a great session. Di, you could have talked for 3 hours. I don't care. Seriously, you can talk all day about this if you want, because in my view, this is like the single most important thing we've heard, principally because it ties together so much. It ties together the Information Commissioner, privacy last night, so many of the things that ethics committees have to deal with. You've really tied together. Yeah, it was wonderful. And I think, it's like it's hitting the nail on the head where ethics committees know there are weaknesses. I have not seen that Essentially Ours document. That actually looks fascinating, because for anyone that ever tried to read Essentially Yours, it was not bedtime reading. And I think it was 3 or 4 volumes, and it is a very good doorstop.
Yeah. Can I just clarify about Essentially Ours? Because I know there are questions there. We're actually in the process of finalizing, and we're actually waiting for final approval from the department before we can release it. But I'm really hoping that we might be able to do that early in the new year. And certainly I'll send it to once we have approval and we have it completed, I'll send it to Gordon so that we can perhaps make it available to everybody.
Honestly, that's just, I think that's just essential. You have to go. So a lot of questions, but let me just start with one. The 2.3.10, the waiver of consent, like, it's a really high bar. If we were to take this seriously, that's just an incredibly high bar. But, sometimes we go, well, it's kind of impracticable. And you know what? Maybe everyone would have said yes. But then some of the ones you didn't highlight were the privacy and confidentiality. And, I just wonder, Di, if realistically, can we as HRECs really get over this 2.3.10 thing?
Yeah. I think I do think it's really challenging for HRECs, and you have to come to your own conclusions based on the expertise that members bring, both from the lay community and from the research community and also the other communities, which is exactly why we have the makeup of HRECs that we do. So you all bring incredible expertise in addressing those questions, but I do still think it's a really big ask for HRECs. And we actually have one of our PhD students is doing her whole PhD on waiver of consent. So we're really looking forward to seeing how that goes.
You can send that one to us. Send that one to us.
Indeed.
But I think it segues into another question. I can't remember who it's from, but it's really about, it's one I had as well, which is about if you don't have the expertise, the national statement says you either have to have or have access to expertise. And in so many of these areas, as I highlighted at the outset, doesn't matter what area of research you want, applications are in, you need that expertise from lived experience groups, etc. Where does one get expertise in this area?
Yes, it's really difficult, isn't it? Because, when it comes to things like waiver of consent, we really, I don't know if there's any one person or body that you could go to assist with those sorts of decisions. We can all bring our own particular views and expertise, but I think we need to have much more discussion nationally on how waiver might apply in this context and how we assess waiver. And, it's something that I certainly would recommend to the new Australian Health Ethics Committee to have a look at it as one of their main tasks for the new triennium. So, Gordon, I think you and I might collectively want to suggest that. And if Jeremy Kenner is on board, maybe he would have some views on this.
Jeremy's online. Hello, Jeremy. I will not say anything disparaging. Roberta's asked, do you have, is it the consent forms are an issue. And actually, just to go back to your schemata thing, one point I had, what I thought was, if someone has already given specific consent for future use, why does it then need to go through an ethics committee again? So, I would contend that maybe, I would say, well, why would you bother if someone's already said you can use this for this particular purpose? But that aside, when we about consent forms and putting text and language that people can understand, do you perceive that is an issue that we have to deal with, given health literacy and genomic literacy? Yeah,
exactly. So I think it's right that if a person has consented and has given consent, clear consent to future use, that's one thing. But as I guess I tried to emphasize, that consent is just one aspect of the process. And the Human Research Ethics Committee just doesn't review the adequacy of the consent, but they review all other aspects of the research proposal. And I do think it's really important for HRECs to review the entirety of the proposal. And often, what we've heard in our research is that members of the public are confident and maybe willing to give consent to future use because they have trust in the Human Research Ethics Committee system that future uses will be properly reviewed as part of the ethics process.
So that's great. So it's a segue into a question from Larissa.
Mm-hmm.
And I mean, you said people have trust in the HREC system. You know, in recent COVID times, they don't necessarily have trust in researchers, but you know, here's hope they have trust in HRECs. What can HRECs do then to engender? Well, can HRECs do anything to engender trust? I mean, HRECs are a function of the institution. So can HRECs do anything, or do we just collectively have to say, well, you know what, we're going to take it upon ourselves to learn more about this stuff?
Mm. Yeah, interesting, isn't it? I mean, it's very difficult to think of ways that HRECs can collectively disseminate what they do.
You can't influence stuff, that's right.
No, exactly. But I think, you know, it's really important to maintain the integrity of the HREC system so that, you know, when participants do participate in research, they know that they have confidence that if they do have any concerns, they can go to the HREC and the HREC will deal with their concerns in a respectful way.
Yeah. And I do think that, you know, I don't like HRECs hiding behind an anonymous wall. Like I think HRECs, you know, have to be there to, you know, provide this relationship. And I know a lot of members won't like that, but I certainly think a chairperson has to be that conduit, because otherwise, how do you know and how can you communicate? And if you just are anonymous and hide, then it makes it difficult. Anyway, enough of my proselytizing. Di, do we need key, this is from Ian Majewski, do we need key legislative change in Australia? Have we got the balance right in terms of privacy safeguards? Who are the world leaders in this area?
Yeah, that's a really good point. I actually didn't elaborate, and I maybe should have done that our privacy legislation is currently in the process of being reviewed. And this happens periodically. There have been many reviews of our privacy legislation, and they've sometimes resulted in legislative change, but not always. Some of the things that are being considered are changes to the definition of personal information, which I think is really particularly important. So thinking about, you know, in the internet era and the era of AI and automated generation of data and so on, the traditional view of personal information as your name and address and so on, it really goes beyond that. So I think analysis of personal information is certainly important. And I think national harmonization is really critically important. It's crazy that we have all these different forms of legislation.
Oh my goodness, absolutely.
Yeah. I would say that I guess the highest level of protection of privacy globally is in Europe. So Europe has these general data protection regulations, GDPR, and GDPR really has a high level of protection. But one of the problems is that it's beastly complicated. So it's hard in some respects to work out what it actually means in practice, particularly in the research context. So we want to have proper protection, but we also want research to actually be allowed to proceed. So it's really finding an important balance between allowing research and, as a consequence, perhaps giving up some of the higher order privacy protections, but making sure that there is still an appropriate level of privacy protection.
So Diamond, I do find that the HRECs, look, I speak my own experience, can be caught in this conundrum of trying to let research happen, but going, you know what, you are not protecting privacy and confidentiality to the extent that we wish. And I noticed you didn't sort of deal too much with a sort of public interest test, and you know, because I sort of feel that there must be some way to engender a view of public interest just to help us get over that line, right?
Yeah, no, I agree. And, you know, I didn't have a chance to cover everything but public interest. And yeah, which is why it's really important to talk to members of the public, to actually really have clarity around what the public interest is. And I think when you talk to people, particularly when you talk in a way that allows people to have meaningful deliberation, people have very much more clear ideas about what they're willing to compromise on to make sure that research can proceed. And we've seen with genomic data sharing, it's really important to be able to maintain those linkages between genomic data and health-related data. So having good security over the data and having trustworthy governance and trustworthy people dealing with the data are really the critically important things.
Yeah, and look, we heard from, as I said, Andrew Killia about privacy yesterday, and Lisa Pomeroy, who's at Metro North here, just about the use of REDCap and things like that. And I think this is just a message for those online. It is about a governance framework. It is about a data management plan. It is about an ethically defensible plan. And these are things that we as ethics committees don't have to do. This is a researcher responsibility. But if they want us to actually agree to the use of genomic samples, then I think we have to set a high bar. Now, Di, 2 more minutes of your time, if you don't mind, because this is just awesome.
I can take 2 more minutes of everybody else's time.
Yeah. Okay, so as genomic research expands, this is from Danielle, can you comment on intellectual property and potential ownership of gene patents? For example, BRCA1, breast cancer in the High Court. How does an HREC consider IP patent and patents in these ethics applications? By the way, but there you are.
Oh my goodness. So this is another area that I am just incredibly deeply interested in. So one of my research themes is intellectual property. And I've looked long and hard over the years about gene patents and the like. This is a really difficult interface, and I guess to my mind it's a really important aspect of governance of genomic data. I'm not quite sure how much it's a matter for HRECs to delve into, but if I could just use the example of UK Biobank for a minute, for example. UK Biobank is one of the big collectors of genomic information and has, I think, genomic sequences from 500,000 Britons now. And they have quite a detailed ethical and governance framework. And one of the things that they say is that, this data should be made freely available. And we expect that there might be intellectual property downstream. It might be necessary to have patents over drugs, for example, to make sure they actually are developed by pharmaceutical companies and they get into healthcare. But they say the UK Biobank says it cannot be done in a way that compromises healthcare. So access and equity are really important considerations. And so they have terms in their material transfer agreements that say this has got to be done in a way that reflects concepts of justice and equity and so on. So it's probably a matter that is relevant to, certainly to institutional arrangements for data sharing and material transfer. So we need to think about what terms should be included in those. I'm just not quite sure whether this is the remit for the ethics committee. I mean, certainly when you're looking at waiver, they have to consider commercial arrangements.
Yeah, I think I agree. I think so far an ethics committee can go to, and we have a lot on our plate as it is. Di, one final comment from David, and one of the other speakers the other day was suggesting that this showed differences in attitudes towards data sharing depending on who will use it. So only 30% supported commercial access. So should consent for further research using genomic data be more nuanced to allow choice between the type of research and who the researcher is? Awesome question, David.
It is an awesome question. And I certainly think that, I mean, these nuances are really important. Researchers and consortia probably don't like those because it just makes it much more complicated for them. But that idea that you can consent to use for certain purposes and not for other purposes, I think is the sort of thing that's showing respectful engagement from participants.
Yeah, so Di, I'm going to stop it there, and thank you all for your questions. I mean, you know, seriously, I think we seriously could have gone on for another hour, but to be respectful of your time and your contribution, I seriously think for me this crystallizes so much about what we'll be talking about on Wednesday and Thursday, and I think, you know, hopefully it helps everyone else just to make these decisions about what we as ethics committees need to do and understand that we have to hold ourselves, and more particularly researchers too, who, as has been pointed out in the chat, sometimes will frame arguments for their own ends, as we all do from time to time.
Yeah.
And please, everyone, please join me in thanking Di. That was a tremendous, it really was a tremendous presentation, Di, and I'm really appreciative of that. And you know, I don't want to be gushy. It's just that it was for me wonderful. So thank you.
Thank you so much. And I really enjoyed preparing and, you know, felt really, as I say, privileged to be part of all of this. So thanks very much, Gordon. Yeah.
Parting comment, and your team is amazing. The venerable Professor Chalmers and Otlowski and oh my goodness.
They're fantastic. I feel so privileged to work with them.
Yeah. I didn't realize there was that. Well, there you go, everyone online. Down there in Tasmania is where you need to go to for your genomic expertise.
Indeed. Yeah. Come on down. Yeah. Thank you. Thanks a lot, everybody. Bye then.
Bye.
Bye.